Welcome to The Explainer. Okay, let's just dive right into this. Today, we are opening up an underground tech briefing to literally explain everything about weaponized sound waves and hacking cancer. We're going to be looking at a theoretical, multi-agent acoustic oncology blueprint. And the idea here is to treat tumors not just with traditional drugs, but with highly targeted, programmable sound.
But we're kicking off this deconstruction by aggressively shattering a massive internet myth straight out of the gate. You've probably seen the claims floating around online, right? The idea that there is some secret magical sound wave out there that perfectly, seamlessly just switches cancer off. Well, our sources make it unequivocally clear there is absolutely no proven universal cancer-killing frequency that permanently flips a genetic off switch. It's just not magic, it's physics.
So the crucial point we need to understand is that if we actually want to use sound against cancer, we must treat cancer cells kind of like a flawed operating system. We have to target their physical stiffness. Scientists call this their mechanical phenotype, which is really just a fancy way of saying how stiff or squishy the cell physically is. We have to focus on that rather than just their biological name, like breast or lung cancer. It is all about how the cell responds mechanically to pressure and vibration.
So here is our roadmap for the explainer today. We'll cover the acoustic arsenal, hacking cell resonance, modulating the gene switch, the fawn and master blueprint, and finally tuning the future.
All right, let's jump straight into section 1, the acoustic arsenal.
When we look at the messy, chaotic reality of ultrasound therapies, it becomes clear this isn't just one simple tool. On one end, at really high intensities, you have thermal HIFU, which is literally burning away tumors through intense localized heating. Then, if you use incredibly high pressure, you get cavitation hystripsy that uses microscopic bubbles to just mechanically fractionate or tear apart the tissue. You've also got sonodynamic therapy, which uses reactive oxygen species, essentially toxic molecules that stress the cell to death. And finally, there are these low-intensity zones that we are going to be looking at very closely today.
Which brings us perfectly to section 2, hacking cell resonance. To really grasp this mechanical failure, we have to constantly contrast healthy tissue with diseased tissue. Think of healthy cells as being relatively stiff and stable. They measure around 1 to 2 kilopascals in elasticity, which is just a standard measure of stiffness. Cancer cells, on the other hand, they are surprisingly squishy and compliant, down in the 0.2 to 0.5 kilopascal range, and they usually feature these abnormally enlarged nuclei. This physical difference, this squishiness, is their ultimate vulnerability.
And this beautifully illustrates that resonance gap in action. Imagine a stiff, healthy cell with a perfectly intact cytoskeleton. It easily resists a standing acoustic wave, kind of like a sturdy building in a minor earthquake. But right next door, you can actually see the softer cancer cell with its enlarged, bloated nucleus, literally rupturing under that exact same low-amplitude vibration. The cancer cell absorbs the vibrational energy, amplifying the stress until its membrane simply tears and it undergoes total mechanical failure. Meanwhile, the healthy cell survives completely unharmed.
But hold on, we need to issue a massive paradox warning right here. Before we get all excited about just vibrating cancer to death, we have to ask a genuinely terrifying question about acoustic manipulation. Can sound actually make cancer grow?
Shockingly, the answer is yes, it absolutely can. Take a look at the YAP/TAZ paradox we've detailed here. If you use a frequency between 0.5 and 0.67 megahertz, you get that cytoskeletal fatigue and selective destruction we just talked about. But if you shift that frequency slightly higher, into the 2 to 5 megahertz range at low intensity, boom! It acts as a tumor growth accelerator. It triggers the YAP/TAZ cellular pathways, which are essentially the cell's biological growth engine, driving proteins into the nucleus and promoting rapid proliferation. It is a true double-edged sword. If you don't tune this perfectly, you get the exact opposite of what you want. That terrifying paradox perfectly sets up section 3, modulating the gene switch.
So as we touched on earlier, sound doesn't just magically and permanently pause a cancer gene like flipping a light switch. Instead, it relies on a biological process called mechotransduction. Ultrasound is a mechanical stimulus that transiently, meaning just temporarily, opens mechanosensitive pathways. This changes the gene expression for just minutes or maybe a few hours. The goal in the lab isn't to permanently turn off the gene forever, but to temporarily open the chromatin, that tightly packed package of DNA inside the cell, making it briefly super vulnerable to immune attacks or co-delivered RNA therapies. Okay, moving right along to section 4, the phonon master blueprint. This is where we start pulling it all together into a theoretical apparatus. What you're looking at is a highly complex, MR-guided, 256-element spherical phased array. I know that sounds like a mouthful, but basically picture a precise robotic bowl made entirely of sound emitters. It features robotic positioning, real-time thermal mapping, and converging acoustic beams that can be electronically steered to hit a microscopically precise focal spot. This hardware acts as the physical hub for all of our acoustic weapons. But here's the thing, the physical transducer is just one piece of the puzzle. Rather than a single clean emitter, this theoretical blueprint is an incredibly messy yet precise 12-agent system. It coordinates absolutely everything. One agent builds a patient-specific digital twin, a virtual replica based on biopsy stiffness data. Another computes the exact resonance window for that specific tumor. Then you mix in the various weapon modes, a phonetic combination engine, strict real-time safety monitoring to ensure you aren't heating things excessively, and finally, immune activation and validation agents. It's a whole symphony of tech working together.
To manage all this and avoid that nightmare scenario of accidentally growing the tumor, the system relies on 5 absolutely critical knobs of tunability. You are essentially treating the acoustic attack like a complex audio mixing board. You have to dial in the exact frequency for depth, the precise intensity to dictate growth versus destruction, the pulse duration, the duty cycle, and even the waveform shape itself. We're talking about using sophisticated audio constructs like chirps, amplitude-modulated carriers, or dual-tone harmonic triplets.
Let's ground all this theory into scientific reality in our final section, Tuning the Future.
I really cannot emphasize this enough. Based on all of our sources, this is not some DIY build you can just hack together in your garage. High-intensity sound can cause instantaneous, irreversible tissue death. Developing this responsibly requires a strict, multi-step progression. You have to start with computational modeling based on the actual stiffness data. Then you slowly move through hydrogels, 3D spheroids, animal trials, and eventually, but only with rigorous ethics and oncology board approval, into human trials. It's a long, careful road.
So, we'll leave you with this final, highly provocative thought to ponder. We know that simple single sine waves aren't the magic bullet, and researchers are actively moving toward combining pulses and chirps and harmonic sweeps. As we learn to mix these complex audio envelopes together like speech formants, what will the ultimate acoustic key to mechanically shattering a cancer cell truly sound like? Thank you so much for joining me for this explainer. Keep asking the hard questions, and I'll catch you in the next one.